from Courtney Thaxton, who's again another staff member at UNC, but is participating today in her role as the co-chair of ClinGen's Gene Curation Working Group. So I will, and I don't think there was any general announcements, so I will hand over to Bill and Courtney. And then, oh, we are recording, perfect.
Great, thank you, Alicia, for introducing us. And could I bother you for access to the screen sharing?
Oh, how odd. I don't have that, Kazan. Do you have that control?
I just shared. You should be able to share now.
It's working great, thank you. Okay. So thanks again, Alicia, for introducing us. I'm presenting this sort of on behalf of like other University of North Carolina kind of biocuration core folks. This idea is the brainchild of my kind of curating mentor, Courtney. So she's going to help me present it a little bit. But the idea that we wanted to share with you today is that the group that we work with at UNC has been developing sort of like a frequently asked questions document for gene curation. And it turns out that a lot of us are coming across the same recurring questions, and that folks that we work with have been sort of collecting the answers. And there's sort of like this living document. I hope you don't mind, I shared the link in the chat so that folks can kind of visit it and see what the document looks like really quickly. But yeah, it's basically like the purpose of like coming and talking today is just for us to sort of get the word out among folks like you that, like us, you know, are encountering these weird gene curation questions, and we want to collect the answers in one place, and some, you know, instructive examples that you can kind of take a look at, if that's okay. And yeah, thanks, Courtney, for pointing out in the chat that the link is in the chat. This is sort of like our frequently asked questions document, and you can kind of go and visit the link over there. Another kind of purpose for today is to kind of consider some of the questions that you all have asked. You helped us out a lot by kind of posing some questions ahead of time, and we're going to try and go through the answers sort of as a group and see, you know, one question that I'm interested in is whether our backgrounds that, you know, coming from different gene curation groups, do we kind of answer these questions a little bit differently? Sometimes there's no right answer for all groups, but there's a right answer for your group, right? And I also want to kind of encourage you, or, you know, ask you a favor to continue to, in that link to the document, to continue to kind of post your questions there. And we'll either answer them or we'll find you the answers if it's something that we don't know, which I think it's likely that there's going to be some interesting stuff like that. So before I get too much into the PowerPoint and stuff, I wanted to just like walk you through the document really fast. This is the same one that Courtney shared the link to just now. But one thing I wanted to kind of point out to you in the document is that we've kind of tried our best to organize it in a user-friendly way. And so it has like a little bit of a table of contents, and everything is linked, so that if you kind of see something in the table of contents, you're like, oh, this is my exact problem. I'm interested in genetic evidence and how to score that. There's sort of like a bookmark where you can kind of like, it'll like link you to that part of the document where we have sort of a list of genetic evidence oriented questions. And in these sections, you know, it kind of like collects all the questions in one place. And then through the bookmarks, again, you can kind of like go down to the answer section. And hopefully, you know, if this is your question and you want to know how to score a family with multiple members, you can kind of like find your answer, find your example, and, you know, links to everything. So that's sort of how it's designed. Great. So again, thank you, Kizan, for kind of like sharing with the group like a survey for collecting questions from this audience. Those were really helpful in kind of giving us something to present on today, and hopefully it'll, you know, the answers were pretty interesting to us, and we kind of like incorporated those into this sort of growing document of, you know, sort of like a question database. And so the first question that we came across from you all was like about sequencing techniques. And basically, depending on how a proband has been genotyped and what kind of sequencing techniques they've been subjected to, how does that kind of influence your gene curation, right? So we kind of thought about what are the different options, right? The genotyping techniques that we see frequently kind of split into two groups, right? There's like a group where you're excited that they were, you know, exome sequenced or something like that, or, oh, great, they have a next generation sequencing panel. So these are sort of the options near the top of the screen. And the bottom of the screen are things that we're sort of a little bit less excited to see. Sanger sequencing of PCR products could be good, could be bad depending on how many genes they looked at, right? Or some kind of like there's a little bit outdated genotyping techniques. Sometimes you'll see that they PCR'd part of your gene and then they restriction digested it to show that there was a polymorphism or something like that. You're not super excited about those techniques. And then there's a couple of things that are sort of in the middle, you know, where there's techniques like linkage mapping or homozygosity mapping that kind of point you to a certain part of the genome that is of interest in maybe one of the families that you're looking at. And then from that sort of like narrow window, you can kind of get further, you know, narrower in the combination with Sanger sequencing. So this is sort of like, in my opinion, sort of a gradient of like really good genotyping techniques from the top all the way down to things that are not super trustworthy near the bottom. And so I kind of drew the line here with like the red, so like the red dotted line, and said that, you know, some of these are kind of under some circumstances could cause you to downscore maybe your genetic evidence a little bit if you weren't trusting the genotyping so much. And there are some caveats here, you know, like even if you have a great sequencing technique, you know, did they focus on a lot of genes in the panel? Are they ruling out alternative causes in some of these other loci? And that could be true of Sanger sequencing too, right? If they tested a whole bunch of genes, like all the important ones, Sanger sequencing could be just fine. So anyways, I kind of took this question from one of the audience members and kind of like made it into question number 18 on our frequently asked questions document. And I think the answer is basically that limitations of the method, that, you know, when you're worried that genotyping, you know, isn't sufficiently looking at other loci, it could be a rationale for downscoring your genetic evidence from that proband, right? And the reason is that other genes that could be causing the phenotype maybe haven't been examined, and they could be contributing to the phenotype. We don't know. So that's a common rationale for downscoring a little bit. And right, and I wanted to mention that there's an exception, right, where when Sanger sequencing has been complemented by another method that really helps you narrow it down to like that particular region of the genome, maybe you don't downscore, right? And another thing that some of my more senior colleagues kind of pointed out is we very, very rarely upscore a variant to say that, to kind of reward a really comprehensive sequencing or really comprehensive genotyping. So this is really like a way to lose points, not a way to gain points for your curation. And so I wanted to kind of like give an example for you to kind of take a look at. And so this is from a recent curation from one of the retina groups. So you can see here, I didn't give you a whole lot of information about the diagnosis, but there's two patients. One is on the pink line and one is on the blue line. And the biggest difference in between them is really how they were genotyped. So in other words, they both have some kind of retinal disease. They both have a canonical splice site variant. It's homozygous in both patients. The biggest difference, right, is highlighted in yellow. One of them was sequenced by a next generation sequencing panel. We know that there's more than 100 retinal genes that were kind of examined, the kind of the best of the bunch. And among those 100 or so, or the one that stood out to the authors was this particular gene, IMPG1. On the other, in the lower patient, they've been Sanger sequenced and they focused on that one gene only, and they happened to find something there. So in other words, the patient on top received a full score, half a point for each variant, or each occurrence of the variant, and on the lower line, we kind of downscored by half, just to kind of account for the fact that there's some uncertainty about other loci that could be contributing. Any questions before we move forward? Great question. Thank you for sharing that. I did want to ask at this point, we've realized that one characteristic that would cause some GSEPs to be kind of more attentive to genotyping than others is having a disease that could be explained by a lot of different genes. So in other words, this example, it's not a coincidence that we shared it from the retinal GSEP. The retina folks have a lot of different genes that could be responsible for similar phenotypes. So they have to pay a lot of attention to what loci were genotyped and which ones weren't. And so I wanted to ask you a quick question. If those of you who are like paying really good attention, if you could kind of put a response in the chat. You know, if you are part of a GSEP that regularly does downscore when you're underwhelmed by the genotyping, could you put like a yes in the chat or a Y? And if you're part of a GSEP that doesn't really worry about genotyping that much when you do your scoring, could you add like a no or an N to the chat? Thanks for participating. It's just kind of a way to get a sense of whether this is a question that has one universal answer, or does it really depend on what group we're working for? Yeah, thank you for putting your comments in the chat if possible. So yeah, it's just like a yes or no question. Do you downscore for Sanger-based genotyping, or, you know, is that not something that you worry too much about? Thanks. So if you don't mind, I'm just kind of following along and looking at answers here.
And I just wanted to note, seeing that I think our chat is about to be overwhelmed by people responding to that question, that there was a question from Konstantinos about what if the phenotype is highly specific, which Courtney has answered for us in the chat. But in case other people were also wondering the same thing, just letting you know that that answer is in the chat.
That's a great question too. Did you want to unmute Courtney and talk about that, or?
Sure. We would not, so even if you have a highly specific phenotype, this genotyping method is still extremely important, because if you did Sanger, you are not ruling out deep intronic splice sites, right? If you are loss of function and you can have a larger range of that introgenic deletion, you would also not find that by Sanger. So it still is better for us when we score it, even with a highly specific phenotype. We're not always sure that the variant that they have picked out is the most accurate one, right? Or all of them that we can assess. So that's why we would still look, even with a highly specific phenotype, at that genotyping and then weigh it for a plus or minus. So really good question. You might be confident the gene is involved. We're just not confident yet. It's the variants that are asserted.
I see what you're saying.
Right. So a highly specific phenotype usually tells us that there's more of a single gene disease relationship. And so the question isn't about the gene, then it's about the variants that you found in the gene. Sanger would not give me as much confidence as having exome or genome for that whole entire gene.
Yeah.
And I think the only other question that asked that I responded to was, what about cases that have validation by NGS and Sanger? Would you upscore? And again, typically we would not use genotyping method to upscore any of it. It gives us more confidence that they've looked at many different things, but then we still need to factor in, again, their phenotypes. How specific is it? What are the functional assays that have been done to show that it's a deleterious or perturbed effect of that variant? So as Bill said, typically it's more of, we look at this as a way to downscore if there hasn't been as many techniques or follow-up experimentation that gives us more confidence in that variant.
These are great questions. Thank you for asking those in the chat. Thanks, Courtney, for kind of like handling them. Sounds really good. Another great question that somebody from this audience asked us ahead of time, and thanks to whoever came up with this one. For X-linked dominant conditions, when we need probands and we find heterozygous females who exhibit milder disease or incomplete penetrance, is it okay to have some probands like that, you know, as part of your genetic evidence? Great question. I have to admit, I've curated a few X-linked dominant conditions and I've never had the opportunity to score a female proband. So I kind of had to research this a little bit, but I asked some of my more senior colleagues and they said, yes, you can score a female proband for an X-linked gene disease relationship, but they raised some really cool caveats. And so I hope you don't mind, I'll share those with you folks. So they said, keep in mind that there's tremendous variability from female proband to female proband, even within the same family, right? And part of it is because X chromosome inactivation can be pretty unpredictable in the tissues that are most important to your disease of interest. And so those who have the wrong X chromosome activated could have really severe phenotypes. Those who have the sort of the wild type copy of their gene activated could have really mild phenotypes. And so they said, keep in mind that that could influence whether you really want to score this proband or not. And so the note number 2 sort of says, you might want to consult your GSEP about this particular female proband you're interested in. Is her phenotype or her features, are they a good fit for your disease of interest? If they're a little bit less severe or less specific to your gene, that might be like a trigger for maybe finding a different proband, something like that. And they also gave a really interesting note to me that you don't necessarily need to have like a detailed investigation of your specific proband's pattern of X inactivation in order to include her in genetic evidence. You can just sort of go ahead and get the GSEPs say so based on her phenotypes. And so I did want to give like an example, like something interesting for us to kind of like sink our teeth into, of like a family where you might want to score the female proband. And so this is a family with X-linked retinal disease, I believe. So this is sort of like an example family, and you can see right sort of dead center in the pedigree is the person who kind of started it off, or kind of the proband of the family that started things, and it is a female. You can see that this is kind of an interesting family because it's a bigger family, but there's definitely like an X-linked pattern of inheritance where everybody's sort of inheriting their susceptibility from the mom side, I guess you could say. At least the males, I guess, are inheriting their susceptibility from the mom side. And I did want to say that you could probably note that some of the females who are genotype positive, and those are asterisked, they are unaffected. So there's sort of kind of like the people were advising me on the previous slide. There is variability in this family among how severely affected the females are. So this is sort of like how the legend, the figure legend for the figure. The shading is for affected folks. The open spaces and open squares are unaffected folks, obviously. Anyone with an asterisk is someone they genotyped. And they didn't really specify among the folks that haven't been, that are sort of like asterisked, but they're open circles and open squares. They haven't specified if they were genotyped, harbored the variant or not. They just said that all the folks that are shaded and genotyped harbor the variant. So I kind of wanted to like ask you this side question, or like a trivia question, right? How many affected people, if you're doing your gene curation record, would you count in this family? And the options are, my instinct before, when I got started, maybe I'll count up all the shaded people, right? Including the folks at the top of the pedigree, right? We know they're affected. Should I like enter them in that box in the GCI? Or is segregation evidence really just asking us about phenotype positive, genotype positive folks? In other words, these folks that are shaded and they have the asterisk. I won't quiz you with this because this is one of those questions that actually does have a right answer. So I don't want to like put anybody on the spot or anything. But the answer is A, right? We only want to focus for segregation evidence. The affected folks that we're interested in are the ones that have been genotyped, right? So you would count 7, at least for me, I count 7 in this figure of folks that have.
There is one exception, because you have an obligate female character between 2 shaded genotype phenotype individuals. That person can be counted for the segregation. Exactly,
So
Bill.
he's counted?
Yeah.
I see. But for the affected.
If it was between a male, sorry. So if the offspring was a male instead of a female down here, that's where your GSEP, you need to talk about how much do they think the female is presenting, and that it is not another variation that is occurring within the family. But that would be typically an obligate carrier. If there is a female unaffected, that's genotype positive between 2 male affected in an X-linked disorder, you could count that as the obligate carrier.
Got it. And so I think you're right. I think there's one on sort of on the right side of the screen too. My cursor isn't working, but there's somebody sort of like halfway down the 3rd generation or 4th generation that she's an obligate carrier too, I think. That sounds good. And so, yeah, I did want to ask this question. This is you could say that this has sort of incomplete penetrance among the females, right? In this family. Should unaffected carriers be listed at inconsistent segregations? We know there's a question about this in the GCI. So just kind of wanted to kind of reassure you folks that these aren't inconsistent segregations just because she carries the variant but isn't affected. That's completely fine. The secondary question that I wanted to kind of run across is, and I think Courtney was getting at this, how many segregations or meioses are present in this family pedigree? The instructions for this that we're supposed to count any kind of parent-child connection or sibling-sibling connection between 2 folks that are genotype positive and phenotype positive. And so I'm going to start with this female here on the left side of the screen. She has a connection to her mom, who Courtney said was, basically, I'm sorry, tell me the term again. She's an obligate carrier? Obligate carrier.
And she's genotyped. So we actually know she's carrying the variant. If she wasn't, right, but was between 2 genotype positive, phenotype positive, that's the true obligate.
Yeah, sounds good. So we do have one of those true obligates. So we're going to count the segregation between this person and her daughter. We're also going to count her segregation between her and her sister, her affected sister, sort of in the middle of the pedigree, who's the proband. You can see we skipped over this affected brother right here because he hasn't been genotyped, so he's not a segregation that we're interested in. The third one is from the proband to her son here. We don't worry about the other son again because he hasn't been genotyped. The proband's other brother is the fourth connection. He has an affected daughter, so she's the fifth. And the affected brother is going to be connected also to this obligate carrier here that Courtney was referring to. That's the sixth connection. She's an obligate carrier because she's a sort of an intermediary between her brother and her daughter's connection also to the sort of the grandson here. So for me, at least, I'm not an expert, but I'm counting up 8 segregations in this pedigree, sort of linking up all the sort of genotype positive and phenotype positive people. And so just like Courtney said, those kind of unaffected obligate carriers do really come into play, and they help us with the evidence because they're connecting up, affected people. Yeah, thanks for putting other cool details in the chat. Looks good. Yeah, great. Right. Any other questions before we get rolling? There's another interesting one after this. But yeah, feel free to put stuff in the chat and unmute if that's easier. The next question I wanted to kind of get into that somebody asked from this audience is kind of another kind of on the same topic, like an incomplete penetrance question. Are there any adjustments that you'd make to the typical procedure for scoring genetic evidence if you're worried that your disease is incompletely penetrant? And so we kind of pulled this pedigree here. I'm sorry, this is like a series of pedigrees, right? I think there's like 10 or so in this figure. And you can see many of them or all of them have penetrance issues. So a lot of times in these particular families, the affected family members look like their children. And a lot of times, I guess all of them harbor the variant in the heterozygous state. So they've inherited it from one parent. But you can see the majority of the parents are unaffected. So we can kind of guess this is a very low penetrance disorder, just judging sort of from these family trees. You can see that in the figure legend, the black, the sort of the dark shading is for early onset disease. The gray shading is for late onset disease. And so, yeah, basically it looks like in addition to the variable incomplete penetrance, there's also sort of variable expressivity, right? There's variability within these families of how severely affected folks are. So yeah, the question is like, how do we kind of navigate this weird situation? Does it make us score the probands less? The advice that I was able to collect from my colleagues is basically we should avoid scoring certain types of probands. The types that we want to worry about are the ones that have a modifier variant that make them sicker than on the other family members, right? So the procedure that you kind of go through is you kind of go through the literature and you try to figure out, are there any modifier genes that have been published? Or are there any cases where there's digenic inheritance, right? Where it's not only my gene, but there's something else that's making these folks even sicker. And we want to particularly avoid probands like that. And in addition, we want to kind of gravitate towards probands that have been genotyped in those modifier loci, kind of ruling out digenic inheritance. In other words, we're kind of looking for cases that are like specific to your gene. The second piece of advice I got is to set a little bit lower expectations for Nomad, right? A lot of times we want to see something, that was a dominant, those are dominant pedigrees I was just showing you. We expect those to be really rare in Nomad if it's a highly penetrant, highly severe disease. This doesn't look like a highly penetrant disease, so maybe we can tolerate some folks in Nomad. It wouldn't be as shocking and you wouldn't downscore for Nomad quite as dramatically. Right. And then what about folks who are part of the family tree, but they don't show the genotype? I'm sorry, they don't show the phenotype. So point number 3 that was raised sort of by my colleagues is you don't worry too much about those folks. You just kind of exclude them from the segregations because they're unaffected, right? They're not necessarily inconsistent or contradictory evidence. So anyways, I was going to come back to this slide and say that we ended up scoring most or all of these probands for the curation that I was telling you about. And the reason is because there were 3 loci that were identified as having modifiers for the gene that we were interested in. And these probands have been sufficiently genotyped to exclude, alternative contributions from those modifier loci. So these aren't digenic cases. As far as we know, these are monogenic cases. Yeah, great point, Courtney. Did you want to talk further about that?
Yeah, it's just another point to always remember in the back of your mind. You're going to read a lot of papers about genes, and it's really important to understand that the current framework is for monogenic disorders. And so part of your task is really understanding, looking at it, have they ruled out a number of genes? If the authors have included that there are other genes known to cause the disease, that may mean you don't score it at all. That won't be a question of penetrance now. That is a question of there's an alternative answer to this. And so that's where you have to balance this low penetrance. Is it truly a low penetrant gene, right? Or is there something else that's actually contributing or causing the phenotype? And so looking at these, you want to rule out those additional genes. That brings us back to the very first question: how good was the genotyping method and how confident are we? And then the other question coming into this, as you said, Bill, where you have several people, a lot of parents that are carriers that are not affected, and then you have their child who's more severely affected, they just would not count, right? Most of these segregations don't meet the qualifications. And if you do have a person that is genotype positive but phenotype negative, you would not count them in segregation at all, because we don't know if they've actually manifested the phenotype. So those are definitely some trickier questions. Hopefully this provides some good guidance, but as we said always, and you might get tired of it, but talk to your GCEP about it, right? Because it can change and alter. And the main thing for anything in these curations, stay consistent. If you have applied a downgrade in one area, you should do it for every single curation in your group, right? The consistency is key, so that you're setting up your own criteria as you move forward, given your particular genes and your disease of interest. Great.
I guess you'd say GCEP traditions or something. I don't know. Just trying to kind of capture the words of wisdom here. So yeah, those are themes that came out again and again as I was talking to kind of more senior folks. Their general advice was, if there's a weird question and you have a doubt, that sounds like a great question for your GCEP when you present to them, and you want to try to stay consistent with whatever traditions they have in that GCEP. You know, if you all always, you know, handle these weird cases this way, you know, try to keep rolling with that. You know, since it'll be something that you can discuss in your publication. Right. So we kind of run out now of the questions that you all had asked. So thanks again for providing those, and thank you, Kaizong, for collecting those. I'm going to go through a couple more just interesting questions that came up from other colleagues in my UNC group that I work with, and hopefully we can sort of like take some votes on some of these that are more GCEP specific, I guess, where the answers are more specific to your experience. So one of the general questions that keeps coming up is, what do we do with these Nomad frequencies? Like, how do they weigh in on the score of this variant that I'm evaluating? It's kind of similar to genotyping, right? In the sense that I don't give a huge upscoring for rarity, but I certainly would like to see it, and I would downscore if there wasn't rarity, right? And so I wanted to give you like an interesting example where there was a red flag. And so one of the really cool groups that we work for is motile ciliopathy. And this is just a case that they came across. And so what they're looking at is basically they have an early onset disease. It's generally severe, and it has autosomal recessive inheritance. So it's not crazy to see heterozygotes in Nomad because it's recessive, right? There's going to be carriers out there. But the disease is pretty rare. It's only like 1 in 10,000 live births. And so the GCEP was really, really helpful to us. They specified sort of like a threshold. And they said, if you see something with a frequency of 0.001 in Nomad, that might be pathogen, that could be disease causing. But if it's much higher than that, they would start to feel really skeptical. So they gave us a number, and they said, above this, we feel weird. And so this variant that we came across in the RSPH9 curation, and I think the curator did not end up scoring anything with this variant. But I just wanted to give you like a kind of like an overview. So when you kind of compare the frequency of this variant in the East Asian population of like 0.008, it's kind of like 8 times higher than the threshold where the GCEP starts to worry. And so the question that I have for you, the audience, like for you, does the frequency of 0.008 prevent proband scoring? Like, would you downscore? Would you not score? And I also wanted to give you like the full kind of backstory on this variant. There's also 4 homozygotes in Nomad. So if you want to put like your opinion in the chat, would you score 0, or would you put low. I don't know if you want to vote for your opinion. Would you score this person at all? Maybe like a yes or no, if you don't mind to put in the chat. If you think
You could
this
also
person,
do a thumbs up, I'm going to score it. Thumbs down, I would not.
yeah, maybe thumbs down says no, I won't, sounds good. So thanks for thinking about this, folks. Yeah, just a quick reminder, it looks like, yeah, severe early onset disease is what we would expect from a homozygote, with two disease-causing variants.
Great, we have one thumbs down, and maybe the rest of you are this. What is the answer, Bill?
I agree. I think thumbs down sounds good. I think you're onto something. Thank you, Sabri. Yeah, it looks like folks are starting to weigh in. Thank you all for participating. I know these can make your eyes glaze over sometimes, these presentations. But yeah, I think you're on the right track. Basically, we did not end up scoring any probands that were harboring this one. We're interested, but it looks, we evaluated them, but no. I wanted to give you another example from the same group, so you can kind of keep your same hat on as far as like, this is an early onset autosomal recessive disease, should be pretty severe, the same threshold of 0.001. We came across this other case that was a little bit more borderline, right? And so in this case, their frequency was higher than 0.001. It was like, yeah, right around that threshold, maybe a little bit higher. The variant, just to give you a background, that this variant was in a gene that didn't have a super lot of evidence of gene disease association, but it was a convincing looking variant. It was an apparent null variant. And I should mention that this is from the Ashkenazi Jewish population, which is a little strange because the group had kind of given us our marching orders of 0.001 as a threshold, but they said that should be for continental populations. So this is sort of a gray area. Would you worry about like a relatively high frequency in a smaller kind of like bottleneck population? And I should also give you the backstory. There's no homozygotes here. Could you maybe like take a vote in the chat, and I'll put like a little marker to say like, this is a new vote. So is this something that you would downscore? Would you score this? Would you do this 0 again? Would you feel more comfortable doing that? Again, this is going to be very much a GSIP specific, probably based on the different experiences that we've all had. And in a minute, I'll give you the answer of what the group decided to do with this particular case. Interesting, kind of a mixture of answers. Some folks are saying score it, but maybe downscore it. There is a question about hypomorphs. Did you want to talk about that at all, Courtney?
Yeah, I mean, I think hypomorphs is an interesting question because, again, this question is balancing the frequency, and now you're talking about frequency to activity. So a hypomorph really means that it has a partial loss of function, right? There's leaky activity that's still there. So it can mean that you might not get as severe phenotypes if you have that in homozygosity or none at all. But as this person mentioned, if you have a loss of function, you may have a phenotype at that point. However, we're still talking about frequency here. So if the presence of that is too high in the normal population, we still have to consider, are we seeing more disease in that population? So you really want to talk with your GSEPs. You may still consider scoring it 0 at that point. Just the same you would anyone else, especially if you're seeing a large number of homozygotes. That leaky activity might be one, but we have the same question with founder variations, right? You could have some pathogenic variants. It's certainly at your group's discretion. And again, balancing at what is the additional details, experiments, data that you have to suggest that this allele is actually pathogenic. I know this is gene curation, but we're still doing a level of this pathogenicity versus could it really be an alternative variant in that gene we haven't discovered, or is it an alternative gene that causes that disease? So this is the balance at the gene level all the time.
Yeah, I agree. It's definitely a balance, and I think different folks reach a different outcome based on how you're weighing the different figures that are coming into play. I love the question about hypomorphs because our group is definitely worried about this, and this is something that came up when we were trying to define talking about this variant. We said, wow, could this be just like a hypomorphic variant that's present a little bit more in Nomad because these folks aren't going to be as severe. And as Courtney said, you really want to take a look at the homozygotes too. If those are in Nomad, boy, it would have to be a hypomorph, right? Because these folks are supposed to be pretty sick.
Or
I
it's
think,
not, or it's not pathogenic at all. And
yeah,
they don't have the right variant, right? So that's where we get back into it. If it was a high number in homozygotes, this is a severe early onset disease, and we're not seeing anything, I would lean towards the more conservative. This is not the variant. You'd have to have an exceptional amount of additional data to show the deleterious effect or somewhat loss of function to do that for gene curation, at least. And you would for variant too.
yeah, I agree. And so the outcome from this group, and this is a very careful group that has a lot of worries about genetic heterogeneity of disease. They said they kind of agreed with a lot of the curators that were weighing in on the chat. They said they wanted to score it, but they didn't want to give it much. So they said, you know, normally a variant of this type, we would give it a starting point of 0.5 point. It's a C-terminally truncating variant that gets rid of about a third of the protein, I think. They wanted to go down to 1 point, just to be careful, because they felt it was getting close to the level that was almost too high to believe it as a cause of disease. One of the reasons that they felt okay about scoring it was that it was from one of the bottleneck populations of Nomad, that they felt a little bit, we don't really know as much about what the founders might be in that population. And also because they felt a little bit reassured that it was only in Nomad in the heterozygous state. They would have felt more nervous if it was homozygous, and then you'd have to start thinking either hypomorph or it's just the wrong variant. Yeah, great question by Beth. If the hypomorphs develop disease later in life, how do you score that? I don't have a good answer.
You have to have a publication that indicates this is the same person and through history they have developed the disease, which often we do not get. So that's another really great point, is we can think all we want prospectively. We can only curate and score for what is currently known. So you do not want to prospectively on any of these classifications think about what could happen. It's what is happening, what has happened, the retrospective, and that's the data we have. We have recuration processes for us to reevaluate. If you are lucky enough, right, to get information on the historical aspect and manifestation of someone's disease, then by all means, right, score that person based on what you've seen through that history and their phenotype. I understand we like to be that way, to think that way, but hopefully that really clears it up for the scoring. At least we have to score right now. We'll do a recuration in so much time later. And there's nothing that would ever, even a definitive gene disease relationship, we cannot say that's it, we'll never rescore it. If there's new assertions for that gene for a different disease entity, we'd have to reevaluate it with lumping and splitting, right? So anything could be at in an opening to be recurated.
Yeah, I think that's an awesome question. And I think that some groups are very aware that the spectrum of disease is kind of expanding over time, that you get the severe cases first, and then you get the hypomorphs later. So I think you kind of nailed it, Courtney, that you used a good word the other day. You said like at this snapshot in time, maybe we can't incorporate the hypomorphs because we don't know enough about them, but we might expect that with recuration, they could fit right in and give us a bigger picture of the disease.
And I mean, we all have to understand that while this is focused on monogenic, we can't say that the rest of our genes and our environment aren't playing a role in why there's different levels of severity across individuals. So regardless, for a lot of different diseases, we could probably see where there are more milder forms. But again, the most severe are the ones that are the ones need to be treated, right? Even more so, that might cause higher morbidity, mortality. So obviously our brain goes to these, but as we're getting more sequencing, we can begin to evaluate those spectrums of the phenotype. So again, this will go into the scoring, and even if you have someone with a variant, right, but if they're not presenting with a phenotype at the time, you can't score them. There's nothing there. Just because they have a pathogenic variant, they haven't manifested the disease. So it wouldn't be incorporated in that as a positive score. It would be 0.
Yeah, Monica raised an interesting point in the chat too, that sometimes we learn about the hypomorphic variants not through the homozygous state, but in the compound heterozygous state with something that's more deleterious, right? That's a good point. That's maybe how we get our foothold into that hypomorph realm, is through interesting combination genotypes.
Yeah, I think the one thing to just be mindful of there is that for that particular disease I mentioned, it can have sometimes late onset. So to Courtney's point, if you expect something to be severe and early onset, then you don't expect the frequency to be that high. But if it's something that has variable onset or variable presentation, it's not incompatible with disease to see something that's higher frequency, but you have to have some explanation for it. And in our cases for Stargardt, there was a lot of published literature for this particular higher frequency variant being in trans with several loss of function alleles in affected individuals at later ages. So one example for your back pocket.
And I think that's why these groups talk about it, right? There's Pompe disease, early onset, late onset. That's the difference between 2 loss of function, right? 0% enzyme activity versus there's retained activity at the end. And you're going to set frequencies, and you could do 2 different ones, but if you set your frequency high enough to account for the fact that there could be a late onset, less severe phenotype, dependent on the type of variation and whatever retained activity, then you'll still be within the range, right? So it's not going to be changes based on that all the time. Most groups are going to set a really conservative, I don't even want to say conservative that way, a more broad range, right? This was a 1 to 10,000, but the cutoff was 1 to 1,000. That might be generous. Definitely go back to your Harvey-Weinberg equilibrium for, right? Autosomal recessive disorders, certainly for those that are dominant or monoallelic disorders, they're going to set those a little bit higher than what the actual prevalence is that they've recorded about that particular disease.
That's raising a great point, that we were looking at a couple recessive examples just now, and you could see the threshold that was tolerated was pretty high. With dominant, you kind of you expect to see fewer of them, right, in Nomad. And so I wanted to give you an example of, this is a really weird example, and maybe it's TMI, but I just wanted to give you an example of a variant that's more rare, but it still gave the group a little bit of pause. So this is a dominant disorder. You can see here that there are no homozygous occurrences of it, but we're not really worried about that. This is a dominant thing. And the frequency in one of the continental populations was maybe 10-fold lower than we were just talking about, but it's still a worry because this is dominant. And so this is sort of a situation where we kind of are worrying about incomplete penetrance. You're kind of weighing like what the incidence is. This is a relatively common disease. It is very low penetrance. And so I'll give you like a really fast forwarded answer that the group in this case is still considering this to be compatible with pathogenicity. It's not ruling it out. It's not high enough to rule it out. And the incomplete penetrance, I guess, was the convincing factor in that case.
And I think the one other thing we want to add to this whole Nomad frequency, if you go back to some of them too, is it's also at your group's discretion, right? Some of these, when we're looking at the highest number, right, that Ashkenazi Jewish on this one, that top number, your groups may also set a threshold for the allele count. Oftentimes, if it's other, you may have a very small allele count that they're just not feeling comfortable on that. So you may have to go to the next one down. So these are just all discussions you should have with your group, right? How many alleles do they want to see for this particular group to be able to really assess the frequency, and that there's not, any kind of bias based on not having enough of a denominator in that calculation.
Yeah, good point. So in this case, we kind of might pick the second one just because it has a higher allele count, feel like more trustworthy. Sounds good. The next one I kind of wanted to get into a little bit was scoring a model organism. I think some of us don't know when a model organism deserves more points, less points. So I kind of wanted to raise this question about, what if it's a unicellular model organism for a really complicated organ level human disease? The default score of 2 sometimes seems too high, right? This is just an example question from the FAQ document that you could sort of like look up the answer if you wanted to. But I was going to share with you some guidance that different groups that my colleagues work for have kind of arrived at. that when you have this, and this Chlamydomonas, I'll give you like a little backstory on it. It's like a single-celled algae, right? So its ability to like recapitulate, you can see on the left side, the human phenotypes for this disease are retinal, they have skeletal issues. None of these are going to be recapitulated by an algae. Kidney disease, things like that. On the other hand, it could replicate some things at the cellular level. We know what the cellular defect is in these humans, is that their cilia are absent or defective. And so what we do see that's a match in the algae are cellular level defects like that. And so I just wanted to share with you that this is just like an example to kind of share. This is a model that received a half a point instead of the 2 default points. And so I wanted to give you like a quiz question, right? So what about this model? So this is like a mouse model. Do you think this is better? I want to ask you if this is better than the algae. Is it worse? Does it deserve the full 2 points? I'm just kind of curious what you folks would think. There's some good questions in the chat. Functional alteration. Yeah, these are great questions. I think there's definitely a point that I want to get to, that some of the evidence that we look at, it seems like it's on the border between 2 categories, right? So a lot of what the FAQ document deals with is which one might be a better fit and under which circumstances you could push it left or right. Would folks mind voting really quickly on this model? You can see on the left side of the screen, it's sort of like a bullet pointed list of what the human phenotypes are. There's about 10 of them. And on the right side of the screen are the subset of those that were recapitulated in the mouse. What would you score this? Keep in mind that the last model that was cellular level only got like a half a point. I think this might be a little bit better in the sense that like the first bullet point is sort of an organ level phenotype. They're saying that there was a reversal of the order of organs in the left to right symmetry of the abdomen, which is kind of a match between the humans and the mouse. That's interesting. And then there's some cellular level stuff in addition. A couple people are saying 1 point. I think that sounds fair. Okay. Here's the spoiler. You are right. The GSEP told us to score this at 1 point. So thank you for voting on that. Let's take a look at the next one. This will be a little bit better. This is again a mouse model. The default score for animal models in general is 2. Question here, what would you score this one? You can see again, the phenotypes are laid out on the left side of the screen. For the human patient, for the mouse, they're on the right side of the screen. It's kind of like a matching exercise. You can see here, instead of like 2 points of matching, we got like 5 points of matching. The strength of the model was considered to be a good phenotypic match. It did have some weaknesses. I guess at the cellular level, there were some differences, like some slight differences, that the mouse was defective, but not necessarily in the same way. Any guesses about this model? Would you give it 2 points? Would you give it 1.5, 2.5, something in that range? Thank you for voting, Christy. You're saying 2. Erica, Laura. I think folks are thinking 2 on this. That is also what our GSEP thought. They said this looks like a 2-point mouse. I'm surprised to see us kind of coming up with consistent answers here, but because sometimes there's such group to group differences, you know. But well done. This is the best one that we've looked at so far. This is a human with a very liver-oriented phenotype here. You can see they have jaundice, they have liver enzymes in the blood, they have, it's difficult for them to move bile out of the liver, bile duct issues, fibrosis. And you can see the match for the mouse on this is quite good. So the mouse phenotypes on the right side, they're recapitulating just about everything. What do you folks think for this one? I think the maximum for a mouse model is 4. Definitely we're going to go above the default on this one. You can see here in yellow, we kind of wrote that the high correlation between the mouse and the human was considered like a rationale for upscoring here. So we're going to go above 2. Laura's saying 3. Thank you for voting. 3.5, 2.5. I think you all are in the right ballpark. The particular group that we work for voted to give this a 3. And I guess their rationale for not going to 4 was that maybe the perfect mouse would have like a knock-in of a human variant. Whereas this mouse is a knockout. They sort of like deleted the whole gene and it kind of matched up with the human phenotype. So they said, well, if you put in a human variant, kind of like inserted it into the mouse genome, that would be a little better than this. So they left a little room to get an even better score. And it sounds like a lot of us are thinking really consistently about animal models. The next question I wanted to ask you, this is a really tough one. I just kind of wanted to go through this if you're interested in. I know a couple people posted questions about evidence that can be on the borderline between 2 categories. And you're like, oh, should I use expression on this? Is this more biochemical? And so one of the crises that people have, that I've sort of come across, I guess, is when I'm looking at localization data, right? Someone has stained a tissue for my protein of interest, and it's giving me some evidence about where it locates to. Does that sound like expression evidence, or does that sound like biochemical evidence? So here's like one example of it. Like maybe I have here like a microscope image coming from mouse reproductive tissue. It's a mixture of different reproductive tissue types like fallopian tube, ovary, other things. The gene we were interested in has a connection to cilia. And so we wanted to know if it was specifically localized to fallopian tubes, where cilia are moving the egg along the fallopian tubes. And that's exactly what the staining pattern looked like. It was not staining ovary, but it was staining fallopian tubes. And so, yeah, is that expression evidence or is that biochemical evidence? The other kind of flip side of the coin, which is also localization data, would more be like subcellular. So like the first evidence was this tissue, but not that tissue, this organ, but not that organ. Another type of localization data could be where in the cell something locates to. Again, we're interested in a ciliary gene. And this is saying that even within a cell, when you zoom in really, really close, it goes to the particular half of the cell that the cilia are on. So the cilia is like kind of like this mohawk haircut that the cell has. And the protein of interest is in green and it's locating to the bottom half of the cilium. And so that sounds more like it's getting at the job of the protein within the cell, right? It's not just saying it's localizing to certain ciliary tissues, it's also getting at the job that it does. So the answer basically to these 2 types of localization evidence is different. One of them is expression and one of them is biochemical. And so here's sort of the answer to that question. Like, here's how you kind of make the determination. If it's a tissue level, that sounds like expression evidence. It shows up in the right tissue. Whereas if it's more subcellular, it goes to a structure like a mitochondrion or a cilium or something like that. That might be more biochemical. That might be like the job that your gene is doing. And so that's sort of like the answer to this question of when you have experimental evidence and it's localization, when is it expression and when is it biochemical? Another question that we've come across sometimes, and this might be the last one that we do, I don't know if anybody has experience upscoring expression evidence. If you do, could you put something in the chat? Could you just say yes or no? If you've ever like bumped up the score on your expression evidence, you'd say, this expression is so impressive, I want to give it more than half a point. Like, I think here, like this is like a typical example of an expression pattern that I might see. I was interested in this gene that had a muscular phenotype. You can see here that the expression pattern has a couple of different peaks. One of them is muscle, so that sounds great. It's pretty ubiquitously expressed in other places at lower levels, and it also shows up in blood where I didn't really expect it. So I gave this half a point. That looks like default to me. But I wanted to kind of introduce to you the possibility that there are some expression scenarios that you come across. They're so darn specific. It might deserve more, right? And so this is a gene ALAS2 that's connected with porphyria, which is kind of like a blood disorder. It has a whopping signal in whole blood. And so it was awarded 1 point. Erica, thank you for weighing in. This is very GCEP specific, right? You kind of need permission from your GCEP to do upscoring on this. And so it's interesting to know that some of the GCEPs don't do that. They don't want to reward that. That's really cool. Do you know if there's like an interesting reason? I'm sorry to put you on the spot, Erica, but is there an interesting reason why they don't like to count expression?
Yeah, one of the reasons is, right, sometimes you don't have enough of the cell type to be comfortably understanding, especially because where some of these genes may localize in certain structures that you're not going to get a really good expression pattern here. So that might be one reason that you talk to your GSEP. Do you just expect to be able to see appreciable or reasonable expression patterns given the cell type that it is? Other times GSEPs don't do it because they just know for some, they're expressed in all different cell types. And for whatever reason, the disease that's manifesting in one organ, but you could still see it amongst others. So they may not necessarily do it to understand. But others on the flip side will say, well, you know, our disease is hypertrophic cardiomyopathy. That's a heart disease. I'm looking at a gene expression and it's only expressed in the heart. Well, they may say, give the highest points you can. It's restricted expression within the organ system where the phenotypes manifest. That might be rationale to do that. But Erica, thank you for adding that, because again, it's really helpful to know that your GSEPs may do things different, and that's not a right or wrong. Again, just making it consistent, which it sounds like they did. We just don't score expression because we can't necessarily trust with what we're observing here.
Yeah, great point. Thanks, Erica, for sharing that. I did want to really quick, I know we're running out of time. I just wanted to do a quick thank you to all the folks that came up with questions for this, all the folks that answered questions when I was panicking. This is the group of people at UNC that does that. Thank you, Courtney, for conceptualizing this good idea. And thank you all for writing in with questions to kind of give us something to chew on and make the document better. I also wanted to mention super fast, just like a quick ad for the Gene Curation small group, which is like a little mini ClinGen group that kind of helped us make this document better. We asked them for permission, to share their email address, which is here on this screen. If you're getting ready to form a new GSEP or you have a newly formed one, you got some questions or some ties to get help breaking. This was an awesome group to help with that. And thanks so much for your time.
Wonderful, and thank you so much, Bill, for presenting that on behalf of the group, and then for everyone, obviously, who's contributed to what is a wonderful resource. So the link for that was in the chat. We'll make sure it's associated with the recording of this presentation when it goes up online as well. And there's been some really good discussion in the chat. So we'll make sure that that's all downloaded and passed on to you as well, so that that can be reflected in this resource too. But thank you again. And we will be meeting again in October, where we will be discussing dosage, I want to say. Now I've forgotten what's up next.
Something informative. So please come.
Yeah.
Thanks, everyone.
Thank you all so much. Thanks for participating.
Bye.
Thank you.